from CAMH in Toronto. The last few years I've been serving on the Ontario Brain Institute Board of Directors, and so I've gotten used to this shorthand of various Ontario institutions. So Sean, maybe you could tell us what CAMH actually stands for.
The Center for Addiction and Mental Health.
Great, thank you. He is internationally recognized for his work transforming our understanding of mental health via the use of large data tools, including machine learning and multi-scale computational modeling. I've been wondering if Sean himself devised his conference as a data source for how the quality of comments entered in the chat varies across individuals in different time zones and at different times of the day. Sean, looking forward more to what you're going to tell us about the role of sleep. Thank you.
So I'll share my screen. Okay. Everybody can see the screen?
Great.
All right. So great. No, I've really been enjoying this this workshop. It's the first time to the BCW, but I certainly will present something I think quite a bit different than most of what you've heard, but certainly with some links to some of the earlier talks. So one of the points, so as was pointed out, I'm at the Center for Addiction and Mental Health. So this is a neuroinformatics center embedded in a hospital, a mental health hospital. And so one of the challenges we're dealing with is, how do we start to understand various mental health and brain disorders? And for the purposes of this talk, I just wanted to highlight a couple common principles that, of course, are relevant to this workshop. First of all, virtually all neurologic, psychiatric, and substance abuse disorders are associated in some way with changes in brain connectivity. And there are a lot of reviews that kind of highlight this. Now, they're also associated with changes in brain excitability. Now, of course, there can be a tremendous diverse set of ways in which that brain excitability is modified. But as Carl was pointing out, of course, this hyperexcitability, this changes in excitability can be a significant factor in psychiatric disorders. And at the same time, if you're from the field of sleep, you know that essentially every single one of these disorders is associated with disruptions in sleep. And many cases, of course, depending on the severity, are associated with some alterations or disruption in cognitive function. And so what I wanted to do today was kind of highlight these areas. Now, this is part of a larger story where I think there are also integral relationships between brain energy metabolism and immune response, but we won't spend much time on that today, just instead focusing on some of the links between circuit excitability, sleep, cognition, behavior, and connectivity. Briefly, one of the examples of alterations in excitability comes from studies looking at changes in somatostatin positive neurons in major depressive disorder, and that's associated because this is a key part of the cortical microcircuitry that regulates the firing of pyramidal cells, and that when there's a decreased inhibition onto these pyramidal cells, you get a hyperexcitability in the cortex, which is associated with cognitive decline and circuit dysfunction. And it turns out that this same kind of marker of reduced somatostatin GABAergic neurons is actually observed in many different disorders, in Parkinson's, Alzheimer's, schizophrenia, bipolar, and so on. So there's a common thread. There is a disruption, at least in this particular type of interneuron in the cortex. And there are other disorders, just for balance, that highlight the impact of other interneurons, for example, parvalbumin positive interneurons. But to explore this, I wanted to draw on the work that I did over some years with the Blue Brain Project. And this is a process, I'm not going to go into any detail here because there's simply no time, but a process of digitally reconstructing cortical microcircuitry by taking the building blocks, assembling them in the volumes of a cortical microcircuit, establishing the connectivity, populating the electrical diversity, the synaptic diversity, and then experimenting on this virtual reconstruction. The key point is this is a data-driven process. from available data and the literature. And these are some of those building blocks I was talking about, the interneurons, the diversity of interneurons in the cortex are on the left, the excitatory cells on the right. And this is the composition. So this microcircuit that we focused on building has about 31,000 neurons, different composition in different layers of excitatory and inhibitory cells. And it's about 250 microns in radius. If it's a hexagon, 230 microns on a side. And there's an extensive process for populating this with those three-dimensional morphologies, establishing the connectivity, validating the connectivity, and layering in, for example, Bouton constraints and so on to establish synaptic locations, synaptic numbers per connection, numbers of release sites, and so on, that all correspond to available measurements from biological data. Of course, the electrical diversity is governed by ion channels, broad family of voltage-gated ion channels. And the process of building the models uses information from the gene expression of the cell types, the morphology, the different types of ion channels, and a machine learning process to optimize the distribution of the ion channels on the morphology to recreate the electrical properties of the neurons. And so this automated workflow is what we use to generate hundreds of thousands of model neurons to build the network. This is one example. So Itai Hai, who is also at the Krempel Center here in Toronto, developed this model of a layer 5 pyramidal cell. Short-term synaptic plasticity is actually very fundamental. As you know, the vast majority of synapses in cortex depress quite quickly, within milliseconds of presynaptic firing. And there are different profiles for the depression and the depressing of the synaptic connection strength. But there are also inhibitory, facilitating, as well as depressing and pseudolinear. So there's different combinations of short-term plasticity that occur on different cell types, both depending on the pre and post-synaptic relationships. And when we put all of that together, we get a cortical microcircuit model that gives rise to these low frequency oscillations when it's generally depolarized a bit. And this is one of the default behaviors of the circuit, just low frequency oscillations. But when we adjust the calcium concentration, take into account differences between the in vitro condition and the in vivo wakefulness-like condition, when we correct for that difference in calcium concentration and the net effect on short-term plasticity, that alters the excitatory and inhibitory balance to produce an asynchronous state that's no longer producing these low frequency oscillations. And then we use that to create a slice that has asynchronous activity that we can do further experimentation on. And one of the key things that we saw is that when you're in a hyperexcitable state where you're basically put the circuit into a highly excitable condition, you get this sleep-like low frequency oscillation. Whereas when you adjust the calcium concentration to represent a more wakefulness-like condition, you actually get a very proportional response. So the number of thalamocortical fibers is along the x-axis, and the response in the cortex is more proportional during the wakefulness, whereas it's a really all or nothing large traveling wave response in the sleep-like state. And that's the hyperexcitability. And in addition, we did some studies to look that there's a very particular balance of excitation and inhibition where the circuit can produce highly discriminating responses to stimuli. And so we could look at the resolution that it was sensitive to and saw that that was very sensitive to this balance of excitation and inhibition. And this is a parameter map then showing for that microcircuit that there's a broad range of states. Some are low frequency oscillation producing, some are asynchronous, and right on the edge between those two conditions, we see this very balanced condition that is most sensitive to differences in different input to the circuit. So just to leave that aside, this is a large team effort, the Blue Brain Project, 82 co-authors on that work. But I wanted to now go and talk a bit about wakefulness and sleep in the cortex. And this is, of course, from Stereata's work, looking intracellularly as well as at the EEG level from neurons in the cat cortex. And this really shows a key difference between wakefulness on the left, which is where the individual cells remain tonic and firing, whereas during sleep, they're interrupted by these hyperpolarizing down states. And there's some interesting aspects of neuromodulation that keep us awake. So there's a whole cocktail of neuromodulators, hypocretin, norepinephrine, serotonin, histamine, acetylcholine, that are released from the hypothalamus, from the midbrain reticular formation. And they basically, this cocktail keeps the brain awake. And when we go to sleep, it's this cocktail of neuromodulators that are being removed. They're no longer being released into the brain. And we start to produce, in this case, non-REM sleep because of this removal of neuromodulators. So in a way, there's this default activity in cortex, which is these low frequency oscillations. And what's interesting is during wakefulness, cortex is actually driven by thalamic input. So if you do, and there's a couple, these two papers, one from Carl Svoboda and one from Massimo Scanziani, where they used optogenetic approaches to turn off the thalamus. And they saw, both of them saw that within 10 milliseconds, the cortex became silent. However, during sleep, if the animal is put to sleep or anesthetized, the cortex starts to produce these slow wave oscillations, these low frequency oscillations on its own. This has also been seen in vitro and in deafferented cortical slab. Now, briefly, these are the primary excitatory cells in the corticothalomol cortical loop. This is from a recent review that just beautifully shows the extent of single neurons and how much they project throughout the entire brain. And these are the different types of neurons that build up essentially the corticothalomol cortical loop. And then there's the Sherman and Guillory model of the thalamocortical loop, which takes into account that cortical areas may be communicating through these transthalamic pathways. And this is where the output of the primary cortical area, layer 1, is coming from layer 5, and then that's actually driving the secondary cortical area. And that there's kind of a hierarchy of subsequent thalamocortical loops that are driven by the earlier thalamocortical loops. And so the very first one is really driven by the sensory input. And this work, which is now at this point very old, actually built a computer model, this is with Giulio Tononi, of 65,000 model neurons, 5 million connections, that really implemented that Sherman and Guillory model of thalamocortical circuitry with a primary area and a secondary area that was driven by the output from the primary area. And this is just to show to scale what the microcircuit that I showed you earlier would fit to. So this is a larger scale, coarser grained model than what I showed you previously. And the model captures different firing types. It's all integrate and fire, different intrinsic ion channel currents, fast and slow excitation and inhibition, short-term depression. It's all based on the cat visual thalamocortical system. And this model we use to recreate the spontaneous activity. It's really just purely driven by optic nerve input. And it produces this spontaneous activity. And then when we present a moving grating stimulus, it's selective for that moving grating. So the vertically selective cells respond just to the vertical component. It produces this onset response. It produces gamma frequency oscillations and a strong offset response. So that was a kind of a model of wakefulness. And then we transitioned to sleep by simulating the removal of these key wake promoting neuromodulators. And what some of the primary effects there are to increase the potassium lead conductance, which hyperpolarizes the neurons, to increase the AMPA receptor conductance, which increases the strength of the excitatory connections, and to increase the persistent sodium channel conductance, which really creates this bistability in the individual neurons. And when we do that, and we go through this transition from wakefulness to sleep, the cells, as they become more hyperpolarized from this increase in the potassium leak, they get quieter and quieter. And then the intrinsic properties of the intrinsic bursting layer 5 cells, plus the persistent sodium, starts to create these oscillations, these up-down states, which are sustained by the synaptic connectivity. And then due to synaptic depression and intrinsic currents, they terminate. Now, the synaptic connectivity is essential to synchronizing this. We do see that individual mini columns within this model can be effectively asleep. And when it only shows up in the local field potential or the EEG, when you have large populations of them actually all going up and down together. So there is this concept of local sleep, which has been observed in many different ways in the biology and in the model as well. And it's really the synaptic connectivity that creates the recruitment of multiple groups of cells into the larger thing, more global thing that we call sleep. And sleep, of course, as you saw in Giulio Tononi's talk from the work of Marcello Massimini, reduces the effective connectivity, in this case, when stimulated with TMS. And then we simulated this with Steve Esser, and this is also work with Giulio Tononi, in an extended computer model based on the same model that you just saw, where we could see that TMS during slow wave sleep actually produced a large response that was not propagated and not differentiated as it was during wakefulness. Now, and final point is that cortical excitability, of course, increases with time awake, and there's a whole homeostatic mechanism around the production of sleep that I won't go into in this first presentation, but it does, the excitability does kind of reset and decrease after sleep. And so that has important implications for the modulation and the functioning of cortical circuitry. And finally, when in this work, again with Giulio Tononi and Yuval Nir, Itzhak Fried, they measured intracranially in patients who had electrodes implanted for either epilepsy or tumor surgery. They sleep deprived these patients and then saw that when they administered a cognitive test, their cognitive performance went down after sleep deprivation, but intracellularly or intracranially, they could actually observe local sleep, local slow oscillations that corresponded in the timing to the cognitive deficits that were observed, and they increased the incidence of these cognitive lapses, and the local sleep increased with sleep deprivation. So with that, I want to thank our whole team at the Krembil Center and our funders, and open it up for discussion. I welcome your questions.
Great. Thank you very much, Sean. So as we're sort of organizing the chat, I just want to ask you a sort of a simple question. Since we know that there are so many sleep changes across the lifespan, and yet, you know, and particularly that as you get older, your sleep becomes more disturbed. Is this a chicken and egg thing? Is there something going on in the brain that's causing your sleep to be disturbed? And then is the sleep disturbance then exacerbating, you know, some of the neuronal loss or cognitive deficits that may accompany aging? Like, how are these causally related to one another?
It's a really great question, and obviously we don't know the whole answer. But of course, we know that plasticity decreases as well with aging, and we know that you know there are many factors in terms of arousal and the release of some of these plasticity promoting neuromodulators, which may also be decreased. So one of the questions is you know, because because it seems, and this is more from the synaptic homeostasis hypothesis of Giulio, but that this plasticity and the consequence of the plasticity is slow oscillations, which may be an integral part of maintaining the health and dynamic range of your cortical circuitry or your thalamocortical circuitry, that as that decreases over time, it's possible that your sleep becomes more and more fragmented because you don't have the global integration and the synaptic plasticity every single day that's going to synchronize sleep broadly across the whole brain. And what I suspect, and there's some evidence in severe neurodegeneration that there are these pockets of epilepsy actually that that are causing some of the psychotic episodes and symptomology in severe Alzheimer's patients, for example. And it may be that there are these pockets of isolated, excitable circuitry that aren't able to really connect to the whole and then become part of this homeostatic process.
Great, thank you for that. I have a question from one of our attendees who does not have access to a mic, so I'm going to read out their question for them at the moment. It says, thank you for your great talk. You mentioned immune system is also involved, but you had to skip it for the interest of time. Could you briefly explain how the immune system is also involved?
Well, so there's kind of a tantalizing line of evidence now linking excitability, mitochondrial stress, and the activation of microglia. And the microglia, after sustained sleep deprivation, microglia are activated and they start to eat synapses. There's a direct link then to this kind of activation of microglia and altering connectivity putatively to regulate excitability, but it's really not clear exactly what their mission is. Microglia are a key part of our development. They're a key part of our kind of maintenance of synaptic connectivity, and maybe they're implicated when something goes wrong and as an immune response. And there's a huge amount of work still to do, but there's some exciting results that suggest that they may be an important part of responding to hyperexcitability.
Again, if I can just jump in here, Sean, because I think it's building on this question I think I'm inarticulately trying to get at, which is this chicken and egg problem, right? So is the inflammation or the immune response causative, or is it a response to something else that might be going on?
I think it's a very core question, but we don't know. And there's a lot of advocates of, well, there's general body-wide inflammation, and that has the causal role. I suspect it can be both, but I do think that there's a very natural progression actually between induced hyperexcitability from potentially trauma or events that you're obsessing over, or there's a range of things that could kind of reinforce and strengthen and create hyperexcitability, disrupt sleep, prevent the downscaling, the homeostatic process, and result in an inflammatory response because of the mitochondrial stress, because of the inflammation triggered by that. And therefore, that's one causal path that there's evidence for, but obviously nothing is proven. And then I think that the other side of it is it's entirely possible that you first have an immune response, which triggers then some activity that starts to prune away synapses and starts to attack neurons and causes issues that way.
Yeah, I mean, this holds interesting implications for the way you would think about treatment, right? I mean, which one is kind of supporting or how can you break these trains? Mac, I'm going to ask you, do you have a follow-up from this, or do you want to ask another question? Okay, then we're going to take a question from the audience. Then Andrea Lupi.
Hi. Thank you for this talk, Dr. Hill. I think it's always very illuminating to see when multiple levels of explanation can be addressed. And in this context, your mention of local sleep was making me wonder whether you think there is something akin to it in terms of a local anesthesia. And I was also, as you mentioned, local epilepsy, it got me thinking that when a patient has neglect, we think of that as a local unconsciousness localized to a particular patch of space. But I'm wondering if that can be turned on its head and be seen instead also as a different kind of perhaps local unconsciousness. And I was just wondering about your thoughts on this matter.
Yeah, I think that's, I think that's entirely possible. I would love to see some concrete evidence showing that is, there's anecdotal observations. There's clearly, I'm not aware of something that's definitively saying that is what happening. I do think that there have been, I'm not going to know the reference right now, but I believe that there have been groups doing local anesthesia for probably in Barrel, as I recall. And then, and I do think that this kind of local epilepsy could very much be kind of creating essentially these effective lesions, right? Effectively lesioning the areas because they're entrained, and of course the synaptic depression would effectively decouple it from other areas.
Thanks a lot. This is super interesting. Thank you.
Great, thanks, John. And that does suggest, again, that maybe we need to move from these kinds of global interventions that we typically use in either for sleep disorders or neuropsychiatric illness to maybe thinking about more localized. Okay, Mac, over to you.
Thanks, Susan. Thanks, Sean. That was a really great talk. And I don't know if you share the same issue, but whenever I do modeling experiments like that, I can't stop staring at the videos. And I end up getting in sort of trouble with my wife for just like zoning out and be like, whoa, look at that. So we've been thinking a lot about microcircuitry, and if there's one rule in biology, it's that everything gets broken. And I wonder if you guys have grappled with some of the challenging issues associated with things like gradients that exist in the brain, right? Where down in V1 or in Barrel, where the blue brain's done a lot of beautiful work, really etching out all the details. If you move, somewhere to the other extreme, say in the cingulate, or if you don't even go that far, let's say just, prelimbic cortex in the mouse, you start to get really odd things like different kinds of projections to different layers, and there's not a really clear layer 4, which is a huge feature in sensory cortex, particularly V1. And I wonder if you guys have kind of grappled with baking in some of those changes across the gradient to see how that might change, the kinds of emergent patterns you see in your models.
Yeah, so the circuitry is in the blue brain models is very much determined effectively by the composition of the cell types, because the morphologies provide a substrate for determining where are the potential connections. And then if you've got morphologies positioned differently in different compositions, you essentially have new circuits, right? And so the circuitry does change very much, as you're saying, across the cortex with this gradient and with this change in composition of different morphological types. And the functional consequences of it, I wouldn't say I have the answer today, but that's at least the approach, and that's how the building works.
Great. So they are kind of constrained naturally by sort of detailed biopsies at different regions. Yeah. Okay, cool. That's great. Yeah, my gut feeling is that there's going to be whole other layers of complexity, second and third order moments of complexity for us to grapple with once we start playing with these things, and then watching them in context as well, evolving over time. There's so much more for us to kind of dig out, but I really love the work you're doing. So yeah, keep it up.
Thank you. By the way, I love your new papers. We're gonna share.
Cool.
So, no,
hopefully we can have a chat about it.
Yeah, it'd be great if we were in person to sit down and chat about it.
Yeah, definitely.
But we should.
Well, thanks for that. John?
Thank you, Susan. Sean, so great talk, and you know, I've seen it many times, but I always get a little bit more out of it. So my question is kind of partly a kind of what's the right way to do computational neuroscience, maybe a kind of methodological aspect to it, but there's also a kind of what's maybe the right way to think about the brain aspect to this as well, which is you know, in the models that you're working with there, and a lot of the models that a lot of people work with, correct me if this is wrong, but I'm pretty sure this is right. You basically, you could probably describe the cortical, you know, laminar circuitry and the thalamic circuitry, thalamic components. These are like structural elements of the model, right? They have, obviously the cell types have their dynamics, but they, you know, the wiring is kind of, you know, it's the wiring is key. Whereas the transition to sleep, right, is kind of neuromodulatory, driven by neuromodulation, right? And it's kind of this this notion that pervades thinking about sleep, and it pervades a lot of psychiatry as well, and a lot of different areas of neuroscience, kind of like brain as a vat of chemicals, right? So right, the difference between the two states is like fundamentally, or a large component of it is the bath of neurotransmitters or neuromodulators is different, and that kind of has a fundamental effect on the dynamics. So my question is, you know, how far do you think that, if you accept my diagnosis, how far do you think that you know, model of the brain in the general sense goes in the sense of like there's no space, there's no structure in the same way to the neuromodulation, right? There's no the wiring of the neuromodulatory pathways aren't really, they're not really there. It's kind of, as I said, it's like vat of chemicals kind of picture of brain states.
There's actually
so,
a
yeah,
new,
if you could kind of riff on that.
So there's it's quite interesting because the specificity of neuromodulators may not be in how they're released, but where they act. And there's a lot of specificity in terms of which cell types and where on which cell types the receptors for different neuromodulators are. And so you may be like taking a shower, but there's only certain parts of you that are getting wet, right? It's highly selective, and it affects different cell types in very specific ways, and synaptic pathways in very specific ways. So I think that the neuromodulators actually are quite key to determining the integration within a cortical microcircuit and how that's going to change, which pathways are prioritized for integration or you know are emphasized in the integration than others, and that changes, of course, with the neuromodulatory condition. Now, it also does seem to be the case that there's more spatial granularity than we've thought. So there's new work mapping out some of the projections for certain neuromodulators, and it's not clear that it's always just a big shower. So I think that remains open to really understanding, and we need these comprehensive maps to understand exactly that grain.
Thank you.
Great, thank you, Sean.
Thank you, John, and thank you, Sean. Now we have a question from one of our participants, Samajita, who unfortunately can't use their microphone. I'm beginning to get the sense that as the evening and night wears on for some of our participants, people are sitting quietly
Right.
while their family members are sleeping. But anyway, the question is, thanks for the great talk. Maybe just a blue sky question. What are your thoughts on using connectivity during sleep as a real trait level measure, since any connectivity during wakeful fMRI, even in resting state, is contaminated by ongoing thought?
I think it's a great question. And in fact, I mean, I think that's why sleep, I think, can be a great diagnostic potentially for network disorders, right? Because it's this point where there's much greater homogeneity in terms of state across the thalamocortical system, at least in principle. And you're really, when you're going into an upstate, essentially all of the neurons, or many of the neurons, are up together, and they're really activating the synaptic connectivity all together. So I think it is a great opportunity to look for signatures in there that actually can help diagnose network connectivity and issues in the connectivity. And I think, you know, also looking for fragmentation in sleep and in different areas that are not being fully engaged. You know, it's clear that after a learning task, you have local areas. This is work from Reto Huber, where they saw increased sleep, increased highly synchronized slow wave sleep over an area that had learned a lot during the day. Okay, so there is this homeostatic and this use-dependent increase in local sleep that is reflective of that day's plasticity. And I think being able to start to see how can we use some of the topography there to help us understand areas that maybe are naturally plastic and have increased slow wave sleep, and those that may be perturbed and maybe not engaged in sleep at night, and therefore maybe pathogenic.
Great, thank you. Thank you very much for that. Okay, and our next question will come from, I'm sorry if I mispronounce this, Fute.
Hi. Okay. Yeah, thanks for a great talk. I'm just wondering, could you comment on how to kind of incorporate the REM sleep into the model, from the transition from beta frequency to you are showing the from beta frequency to a theta frequency, and I was wondering if how to incorporate the REM sleep into the model.
Sorry, I had a little difficulty hearing exactly. Susan, were you able to? No.
Oh,
it's a little bit hard, and I wanted to be okay for REM sleep.
Yeah, basically.
Okay.
Yeah.
Okay. That's a great question. So these pedunculopontine projections that basically drive cortex and thalamocortex during sleep. You know, it seems that there's tonic firing in these nuclei, which kind of drive a depolarization and this activity in cortex. And it would be really great to integrate that into the model. I don't have enough data specifically about what they're doing, and I would love to. You know, I think it would be very natural. It'd be very fascinating to model that and to try to understand, in particular, what the specific neuromodulatory effect is, and also what the pattern of firing is doing on the thalamocortex. But at this point, I think we still need some more data to understand how to model that accurately.
Thank you.
Great, thank you. And our next question will be from Mary Pat.
Hi, Sean. Thanks very much for the talk. Really wonderful. This kind of follows on a previous question, but we've been doing some work trying to understand sleep and memory consolidation. And one of the things that we found in looking in patients with temporal lobe epilepsy in particular, and interestingly, unlike healthy controls, where usually you find a positive correlation between time and slow wave sleep and overnight consolidation and memory performance, we find the opposite. In fact, a negative correlation. And we know that spikes are more likely to occur in slow wave sleep. So I was kind of wondering if you could kind of give me some insights into what you think the impact of kind of local hyperexcitability might be on this hippocampal neocortical connectivity that we think is important for consolidation.
Well, so it's a really great question. I think one of the striking things is that when, you know, and particularly in the case of epilepsy, when you go to sleep, there is actually kind of this increase in synaptic connectivity. So if you have maybe something that doesn't show up as epilepsy during the day, it could be more, and of course, as you know, with epilepsy, if you're sleep deprived or
Right.
you're more likely to have a seizure, but it could be that internally something becomes more, epileptic at night, or is at least to a level of activity that prevents the homeostatic plasticity to restore essentially the balance. And so it could actually be interfering with what in a normal healthy individual would be a consolidation process, because you're actually downscaling synapses, if this synaptic homeostasis hypothesis is correct, but rather pushing you to an excitability level where that plasticity cannot take place. Actually, you're pushed out of the realm of
Right.
the plasticity, and we know that that's there's a very particular level of for NMDA receptors to be effective for plasticity, and as well it's a window, and if you're pushed beyond that, it's conceivable. Of course, we still need the evidence, but it's conceivable that you're being pushed out of the realm of the downscaling plasticity.
Right. Are there any measurements that we could be able to take that would, in healthy or in humans that would allow us to have a look at that?
It would be interesting to talk more about that, but looking at like the slope of the slow waves in non-REM and seeing if there's any kind of change in that slope.
Mm-hmm.
And is it particularly high at the beginning of the night? It'd be useful to kind of see, because that's kind of an indication of how much the neurons are being recruited simultaneously,
Mm-hmm.
and whether or not that differs from kind of a so-called healthy slow wave. If it's much steeper, it would be very interesting, and then it would be of course interesting to see the evolution over the course of the night.
Cool. Well, we may trot down from the Toronto Western with
Great.
some data.
Be great to talk with you.
Great. Thanks.
Thanks.
Okay. Next up is Dan. Laurie, go ahead, Dan.
Thanks for a really, really great overview of these modeling studies, Sean, and also just kind of the degree to which we kind of start to get an idea of what's happening at these very local population levels. And I'm wondering, so you talked a bit about the idea that neuromodulatory tone is kind of what this big shift in global brain state from wake to sleep, and also we know that there's kind of this idea that maybe that happens at a phasic basis locally during like attention due to acetylcholine. And I'm wondering if you have any thoughts about how the state of kind of cortex maybe projects then back down. So you're talking about the idea that like EI balance kind of changes throughout the day, and that like with wakefulness, and maybe in like the extent like is there a method whereby that then kind of feeds back down to these nuclei to kind of turn off the bath of neuromodulators to say, okay, now we're going to go back to a more synchronized sleepy state?
Right. So actually, there's a lot of attempts to look for that sensor, right? What's that sensor that tells you when you need to go to sleep? And of course, one component is the circadian cycle, which is driven in part by the suprachiasmatic nucleus, and is one signal, right, for sleepiness. But then there is, of course, the homeostatic pressure, the sleep pressure. Now, there was recently a work published by Vlad Vyazovsky, where they were claiming, and I need to read the paper in detail, but they were claiming that it's the intracellular chloride potential that actually accumulates in neurons and kind of creates this sleepiness signal. I don't know. I don't have an answer for you in terms of what definitively does it, but if you think of throughout the course of the day, excitability increasing, there's the potential for so many redundant mechanisms to be that signal, right? Both in terms of voltage and voltage sensors, and as well as mitochondrial signals and alterations in ATP production. There's a whole range of things, but but I don't have a clear answer on that.
Well, thanks.
Great, thank you, Dan, for asking that question. And Mac has another question he'd like to throw at you, Sean. So go ahead, Mac.
Great.
Hey, Sean, yeah, I can't wait for my turn. I think I'm just excited. So you did some really beautiful modeling work down at that little micro scale level, but a lot of people have made assumptions, Gaussian assumptions, and tried to kind of zoom out to the sort of mesoscale level and do things like neural mass modeling, neural field modeling to good effect in many cases. And I wondered if you were feeling towards the end of your chat in a speculative mood about what you think the kind of links are there, and whether or not you think there are kind of sensible ways for us to do mean field modeling that are kind of more respectful of the kinds of detail that you're seeing down the microscope.
Yeah. So I think there's a lot of opportunity there. I think that one of the challenges is of course also in the coupling between the populations and of having a good model of the short-term plasticity that links the areas. And a lot of the mean field modeling doesn't per se have that, in the sense that it's going to depend on the individual population's presynaptic firing rate, and then some populations facilitate, some depress. And I think taking into account and kind of respecting some of those rules of the short-term plasticity would be very interesting. And in particular, in the generation of sleep, I think very often models of sleep end up building these kind of coupled oscillators that, one is kind of suppressing the other, and then they're producing the up-down states through either inhibition or through some form of coupling. And I think having this, that you can still evoke a response. It's not as strongly inhibited, it's a disfacilitated state, and that input can still get through. And therefore you have this very different nature of how things can synchronize across different areas. And I think that that's an important piece as well. I'd certainly be interested in talking about ways in which the neural mass models could better capture sleep. And I think that's an important area.
Great, thank you. We have another question from John Griffiths. Go ahead.
Hi, Sean. And, people who know me, they might imagine that I would be asking a follow-up to Mac's question, because that's exactly the kind of thing that I'm interested in. But I have a very different question. So, one of the interesting things that's going on in, outside of the small science world that we live in is that, sleep tracking is kind of working its way into daily life for like large numbers of people with wearables of various kinds, and one of the things that comes with that is things like even polysomnography visualizations. Like people can get like email reports from their Apple Watch and their Oura Ring giving them a plot of their PSG kind of sleep stage transitions. That it's kind of, well, firstly, it's just interesting, kind of crazy that that's something that like large numbers of people have access to, notwithstanding its quality. But question is, with that general trend in mind, right? Like we definitely, wearable devices are coming through, and we know there's information to be had there. Like if you were to take your physiologist, computational neuroscientist hat and say, well, what would I want to know if I could get a kind of like wearable tracking of the state of the circuit simulation that you were showing, or the physiological characteristic of that? What numbers would you really want to track and maybe output in a fancy graph and kind of let people know about? Can you kind of think where you would go?
Yeah, I mean, at one level, I think one of the big challenges is, and this is something that we saw in the modeling, is that at the EEG level, the real state of the system can be masked just by watching EEG, and that you need to do a perturbation in order to reveal the actual state the neurons. And this is, of course, something, you know, Marcello Massimini has developed as a way of really assessing excitability, you know, all around the brain. But unfortunately, TMS and TMS EEG remains remarkably coarse and very limited in terms of where you can actually probe. And you know, I would love to see, you know, how could we measure at a finer scale? How could we perturb at a finer scale? How could we measure the excitability in terms of that response to perturbation at a finer scale, to be able to better map out the disrupted excitability all around the brain? And I think that's one of our big challenges, is we don't have those tools, and the EEG alone is you know, very likely. I mean, there certainly may be signatures in there, and I think we have to keep looking. We have to keep trying to find, but it's quite challenging, just because of the nature of the circuitry and the way that it can continue to flow through different forms of activity and oscillations that don't really reflect in a precise way the actual state of the underlying circuit.
Great, thank you very much. Sean, I'm going to take the chair's prerogative and ask you sort of the last question before we transition to Mac. So, and it sort of follows up on John's question a bit, in that we are acquiring enormous amounts of data, but even never mind this you know, everyday user kind of data, which we know has uncertain quality. But in the data that we do have that's good quality, you raised some interesting questions about these diagnostics. So is there information that we are tossing away because we're not looking at the right part of the data? Like, we're not paying attention to what we really should be paying attention to? And do you have any suggestions of some things that we might want to be looking at a little bit? You mentioned in the slope and the slow wave sleep. I think that's a really interesting idea. Are there other kinds of things that we should be paying attention to that we may not be?
I think the topography, you know, is something that can certainly be revealing, right? In combination with the temporal resolution, having increased spatial resolution obviously is going to help. But I think, you know, seeing where there may be something disrupted, or where the propagation of something does not pass through. For example, you know, a slow oscillation, if it's propagating, is it disrupted by something? So that topography, I think, can be useful. I think we need to learn more about, you know, what is there in the spectral space that could give us more information. You know, obviously slowing in the EEG is something that's been remarked on as a sign of aging and as a sign of dementia and so on. But what more precisely? And the slope work really comes out of the, you know, synaptic homeostasis hypothesis and the work there done in across different animals and in computer simulations, as well as in human. So that was kind of one of the things that stood out there. But I think we need to continue to look for more precise things. I know Julio is there. I'm sure he has some ideas.
All right, great. Julio, did you want to say anything before we close out on Sean's session?
No, I'm fine with this. I happen to be Gustavo. I don't know why, how that happened. And so throughout, I was listening as Gustavo, and like Gustavo, I will shut up.
Well, thank you. We're happy to have you in whatever guise you appear, Julio.
Incognito.
Okay, so with that, we will move to our final talk of this.